Hi, this is Sonal and Emily, and today we will be discussing the article Hepatic ADTRP Overexpression Does Not Influence Lipid and Glucose Metabolism by Dufour et al. Just as our cells contain receptors on our plasma membrane to perform specific functions in our cells, there are also receptors on our nuclear membrane that allow for particular physiological responses. Peroxisome proliferator activated receptors, also known as PPARs, are one such type of nuclear receptors. Ligands such as fatty acids, environmental contaminants, or drugs will bind to these receptors and cause them to translocate inside the nucleus itself. Here, PPARs will bind to particular sequences on the DNA called PPAR response elements, or PPRES. PPARs act as transcription factors by causing the transcription of certain genes. Most of the target genes of PPARs are involved in lipid and glucose metabolism in the liver and other tissues. This can be seen in this figure over here, as fatty acids and other ligands will bind to these receptors that exist on the nuclear membrane. They will translocate to inside the nucleus and bind to certain sequences on the DNA itself. Co-activators will cause them to transcribe certain genes that relate to lipid metabolism within the liver. In this paper, we'll be focusing on one particular nuclear receptor within the PPAR family called PPAR alpha. This particularly regulates lipid metabolism within the liver. A purpose of this experiment was to determine the novel target genes of PPAR alpha. It was found that there is a strong binding site for this transcription factor in the immediate upstream promoter of the ADTRP gene. This can be seen over here as PPAR alpha has multiple effects within the liver itself.
Fatty acid esters of hydroxy fatty acids, also called FAHFAs, are composed of a fatty acid esterified to the hydroxyl group of a hydroxy fatty acid, and these have been identified as a potential insulin sensitizing class of lipids. These are hydrolyzed by various enzymes, one of which is ADTRP, the gene that is targeted by PPAR alpha. ADTRP derives its name from androgen-dependent TFPI regulating protein.
So in this first set of figures that we'll be talking about, ADTRP mRNA levels were measured in both mouse primary hepatocytes, which is shown right here in figure B, and in liver slices as well, which is shown over here in figure C. Right down here where it says CNTL, that stands for control. So in these cases, samples, so either the mouse primary hepatocytes or the liver slices, were just placed in a normal medium. In these samples over here, where it says WY14643, that is an agonist for PPAR alpha, meaning that either the mouse primary hepatocytes or the liver slices were placed in a medium that contains supplementation of this particular agonist. So as this agonist will increase the activation of PPAR alpha, the purpose of these experiments was to determine the impact of increased PPAR alpha stimulation on the expression of ADTRP. Supplementation of this agonist within the medium clearly led to an increased expression of ADTRP's mRNA. This can be denoted by 3 asterisks between mRNA expressions of ADTRP between the control and the agonist supplemented medium. These 3 asterisks show that the p-value after doing a t-test comparing these 2 samples was less than 0.001, indicating that there is a great statistically significant increase in ADTRP mRNA levels when there is increased PPAR alpha stimulation by this agonist.
So, although the aforementioned figures demonstrate that ADTRP is upregulated when PPAR alpha agonists are present, this upregulation does not lead to metabolic effects in the mice. Even when ADTRP is overexpressed, total liver triglycerides, as well as liver and plasma FAHFA levels, were not significantly affected. So, figure 4 on the slide is a volcano plot, meaning that highly statistically significant data points having low p-values appear towards the top of the plot. It is clear in this figure that most points are towards the bottom, meaning they are not statistically significant. This test was performed in the liver, and the lipid levels were not significantly impacted. This allowed the researchers to conclude that there are not significant metabolic effects of ADTRP overexpression. So after determining that total lipid levels were not impacted by upregulation of ADTRP, the researchers tested levels of 4 specific types of lipids, as seen in the figure: triglycerides, cholesterol esters, diacylglycerol, and phosphatidylcholine. As can be seen between the GFP samples, which are simply control samples with a fluorescent protein added to make it easier to measure lipid levels, and the ADTRP samples, there is no significant difference in any of the 4 lipid levels. This can be determined by the fact that there are no asterisks present between the GFP and ADTRP samples. So some conclusions that were drawn from this research: PPARA, a specific type of PPAR, is important in liver metabolism regulation. ADTRP is a gene target of PPARA. Experiments showed that upregulation of PPARA led to increased ADTRP mRNA levels. However, upregulation of ADTRP expression did not actually impact hepatic lipid levels. Therefore, ADTRP may not have a clear role in actually contributing to PPARA's function of liver metabolism.
And finally, we have 3 questions for you guys to answer. So number 1, if FAHFAS are a potential insulin sensitizing class of lipids, how does this relate to type 2 diabetes? Number 2, if ADTRP expression increased with a PPAR alpha agonist, how may the expression of ADTRP change with a PPAR alpha antagonist? And number 3, the article attempts to see if ADTRP has an impact on lipid digestion in the liver. What substance did we learn about in class that helps aid this process?